welcome everybody here. I'm really happy that so many of you showed up. It is great pleasure for me to welcome Patrick Cramer here to deliver the MENDEL lecture. I will say a few words about him because Patrick and we go quite a way back. We started our PhD together in 1995 at EMBL, in the famous EMBL PhD program. I finished my PhD at 2000. Patrick Cramer finished his PhD in 1998. So it's the sign of the things to come. Patrick is a chemist. He came to EMBL already with quite some experience with science. We overlapped for a couple of months during a PhD course, and then he did all his research at EMBL Grenoble, whereas I stayed in Heidelberg. So Patrick is a structural biologist, and back in the days he was primarily using crystallography to study the structure of proteins, and he got interested initially in the structure of a transcription factor, NF-kappa B, of which he solved the structure thanks to a trick, which, as he told me, he rediscovered. It's a trick to shrink the crystals. Maybe the expert in the field know about it. Then he moved on to a postdoc at Stanford, to the laboratory of Roger Kornberg, where he solved the structure of something much bigger. And it's nothing else than the RNA polymerase. This work has been foundational for our understanding of biology. And Roger Kornberg was awarded a Nobel Prize for this work, which Patrick performed with his hands in his lab. We are talking here about 2000, roughly, right? So at that time in Germany, it was customary for German scientists to go to America and then come back. In German, it's called in America gewesen. So Patrick did that, and then he was recruited back to Germany when he started his research group at the University of Munich in 2001. In his career, he has reinvented himself several times. His first reinvention was to expand the repertoire of techniques which he is using to study transcriptional regulation to include genomics. In 2001, he started his lab at LMU. In 2004, he became a director of the Gene Center in Munich, and he ran this institution for 10 years. Then he was recruited by the Max Planck Society, where in 2014 he became a director of a department in Göttingen. Around that time, he reinvented himself one more time, and he included in the repertoire of his techniques cryo-electron tomography, the single particle kind, to study transcriptional regulation. Patrick will tell us a lot about that. His vision is to provide us with a really dynamic movie of how RNA polymerase transcribes genes, and I think he comes very close to that. Personally, what I would say is that in his career he's been incredibly productive. So whenever I opened a glossy journal of your choice, maybe it's called Nature, maybe something like that, and you see something about transcription, you kind of think that's going to be a Cramer paper. And you find out, indeed, that it's a Cramer paper. So, the third reinvention of his career is really kind of dramatic, and I have a personal story to tell about this. At the time when Patrick was recruited to the Max Planck Society, I was a representative of the Dresden Institute, and I was coming to the meetings of the society, and I could see immediately that Patrick definitely belonged to the Max Planck Society. He was very active, and he was, a go-to person for many things which the society has been developing. For example, the famous Perspektive and Commission, which is looking at where the society is going to go in the future. So this was probably around 2014, 15. I was telling to my friends or to people who would listen at that time, one day Patrick Cramer will be the president of the Max Planck Society. And in 2023, this actually happened. So he is now the standing president of the Max Planck Society. He has very good reasons for that. He hasn't lost his passion for science. We have seen that today through the numerous discussions which we had about science. The passion is still there, and you will see it during his lecture. I think that his decision is really motivated by the desire to help the society. And I think he's really the right person to do that. I think we cannot, the Max Planck Society cannot wish for a better president because he is one of us. And I say that because I know him, and I think it's absolutely true. So that's all I would wanted to say. Patrick, of course, throughout his career has gotten many prizes, and he doesn't want to talk about that. But I just have to mention that just today he was elected a foreign member of the Royal Society. So congratulations, fantastic. And last but not least, as we were walking around SATEC and looking at the various instruments which are available there, Patrick spotted an electric guitar on the wall, and I learned that he is in fact electric guitar a player, or used to be. Now he more or less collects them. He has quite a few. They have some special names, I forgot them, but he seems to be actually quite passionate about it. So I think he will not play the guitar here, but he will give us a lecture about the transcriptional regulation of the genome. Thank you for being here. Now, I'm sorry that I kind of start sounding like I'm wrapping it up. I am wrapping it up, but it's my honor also to present you with the MENDEL medal. Right? So no, not MENDEL, but MENDEL medal, right? Which is, goes to everyone who gives this MENDEL lecture, and we are really happy that you are here and looking forward to your presentation. So I will now give you the medal and we take a picture.
Thank you. Thank you very much. Amen.
And this certificate which comes along with it, I can hold the certificate, you hold the medal, and we take a picture.
Okay. Thanks. Give this to my wife. Well, thank you so much, Pavel. It's wonderful, very kind of you. And thanks everyone for coming. It's very moving to be here because for many years I was here a long time back and I didn't have time to come to this place. And now I had the time, so it's very moving. And why is that? It's because when you spend 30 years of your life in molecular genetics, right, and then you come to the place where it began, it's incredible. I could wander around here for days. I had a great tour. And there's many exciting things about Mendel, but 3 things clearly stand out for me. The first is he followed his curiosity. You see the phenomenon of inheritance in nature and you want to know, how is that possible? What is the rules? What is the underlying mechanism? Second thing, to solve this puzzle, he had to cross borders, and he even had to bring 2 disciplines together, right? Botany and statistics. And I think that's probably why he wasn't understood at the time, and it took 2 generations, 35 years, until his work was rediscovered. And that brings me to the third point. In science, sometimes it's frustrating, and sometimes you need to be persistent, especially when you're at the forefront. When you do mainstream, you just contribute more details to a big research field. But he invented a new field at the interface of botany and statistics. And that can be a lot of frustration. So I learned that he sent a manuscript also to Darwin, his manuscript from 1865. And he didn't even open the mail. It was later found after Darwin had passed away unopened. And it took until 1900, 35 years later, until his work was rediscovered. And then he was realized as the founder of a new field, genetics. So I think it's also the beginning of biology to be quantitative, to be a modern science, if you wish. And in that respect, it's a pleasure to give this talk about transcription, which is, if you wish, part of molecular genetics. And the work was done by a lot of people. You see the list here. And you also see that most of them are listed under past, and this has to do to some extent with what Pavel just mentioned, namely that I took office as president. It was a difficult decision, but I don't regret. It's very exciting. It's a new type of job. But of course, my heart still beats for the science. And there's also still a team present at Göttingen with many good people, and the young people are now obviously taking over. By the way, those who are here in color, they run their own laboratories. So over 20 people actually went on and had their own research groups. And this is a great joy for me, but also a relief in the sense because I know that the field will be carried to the future. This is the issue. Mendel went to the garden and he spotted this phenomenon of inheritance, right? The colors can change, they can come back and so forth. And in transcription, I think the paradigm is how can you go from a fertilized egg to an organism and make all these different cell types. Each cell has the same set of genes. So how is it possible? It's very simple because in certain cell types only a subset of genes is active and the other genes are repressed or switched off. And this is what we call transcriptional control, right? And over the last decades, my passion was to understand those switches. How do you switch a gene on or off? It turns out there's not one type of switch, there's actually several types of switches. And hopefully at the end of my talk you will have learned a little bit about how these switches actually look like and how they function. First of all, what you need to know is that there's 3 enzymes in our cells that can transcribe genes into RNA products. And together these RNA products are then used to make proteins. And of course, when you have different types of proteins in your cells, you have different functions. And this is basically underlying gene expression. I was always most interested in RNA polymerase II. We also worked on the other polymerases. And the reason is it makes messenger RNA. And the messenger RNA, as you know, carries the information on how to make a protein. But you also, of course, need the other RNAs. And in my career, we went from black boxes where we had no ideas how these RNA polymerases work. We didn't know how they look like, to atomic structures of these enzymes. Very fascinating. And then, when you have even several snapshots of these enzymes in different functional states, you can even make a movie. And this was a big breakthrough after about 10 years working in our lab in Munich. We went to the first movie of gene transcription, and I wanted to show it today because it's still fascinating to see. So in blue you see the DNA template, which serves as a template to instruct RNA synthesis. And in red is the RNA product. Here's the active site. And now the next nucleoside triphosphate substrate is going to enter in orange. This is a substrate, and it's Watson-Crick base pairing, and the active site is closing, and then you form a phosphodiester bond. So the RNA is now extended by 1 nucleotide, and the enzyme undergoes a conformational change in order to translocate and bring the next template base into the active site of the polymerase. And then this cycle that is called the nucleotide addition cycle can start all over. So it continues like this. So that was nice to have a movie, and that was, in 2010, 2012. So we saw, how does the enzyme actually make mRNA, how does it use the gene as a template. But of course that doesn't tell you anything about how genes are regulated. It was nice because the black box was opened and we could see how genes are transcribed, how mRNA is made. But the next big challenge, of course, was to understand regulation. How do you regulate an enzyme like RNA polymerase with 12 protein subunits? So the answer is that about almost 10% of our genome encodes for transcription factors that actually regulate RNA polymerase II. have to think about that. You have an enzyme that copies DNA into RNA. Now you want to regulate it, and you come up with 1,600, maybe 1,800 proteins to regulate this machine. Why do you need so many? Actually, it turns out this has to do with the multicellular nature of our organism. So when you look in bacteria, you have far less of these. You also have transcription factors, but the regulation is, less complicated. You need to regulate, for example, when there's changes in the environment, nutrition, temperature, and for this you have transcription regulators. But if you want to move from a fertilized egg to an embryo into a full organism with hundreds of different cell types, then you need 1,600 or even more transcription factors. And in these different tissues, if you just look, which transcription factors are active in these different tissues, you see that different transcription factors are active in these different tissues. So depending on which transcription factors you express, different types of genes are active. And now you see a very important principle, namely what I said in the beginning, that you switch things on and off. It's not that simple. And why is it? Because the switches are under the control of the machinery that they switch on and off. So you see, you have a regulatory circle. The products of the genome are regulating the genome. First insight. That's actually why, when some people think, sometimes they come from physics or so, when they think, oh, we can make a cell by just putting the genes into a box and then providing all the, metabolites and so forth, and then maybe something happens. Nothing happens. And why is that? It's because you need life to make life. We are all connected to the last universal ancestor, everything that lives. So this flame of life has never been extinguished, has just been handed over and over and over. And this is part of the reason, because to switch genes on and off, you need already the products of the genes. Where do they come from? Right? You have them in the fertilized egg, you already have a set of them, and then you move from there. Now, how do you even dare to study such a problem of gene regulation? It's so complicated. Certainly, to get the structures, like the movie I showed you, if we would have more movies of how the gene is switched on and off, that would be nice. And we have some by now, I'll show you some. But this is not enough, because obviously you need to also be in living cells, not just in the test tube. So you need to combine the in vitro biochemistry, structural biology, with in vivo genomics. Pavel alluded to it. This was a major step for the lab to do both. And then when you have both in the lab, you can actually go forth and back. So you can, for example, generate a hypothesis using your structural biology, and then you test it in living cells. You test your hypothesis. Or you take data in living cells and you generate a hypothesis based on statistics, like Mendel actually did for the plants, and then you test it mechanistically in the test tube. So if you have both, you can go around in circles, and it's actually not a circle, it's actually a spiral that goes up like this. So the knowledge increases by going forth and back in these cycles between a hypothesis and then a test of the hypothesis, improved hypothesis, and so in the end you can increase your knowledge of the system. Now, a great colleague of ours, John Liss, published 15 years ago in a journal, what is it called again? Nature. This nice review where he said, regulation is mainly at the beginning of transcription, but the beginning, the initiation and initiation elongation transition is actually quite complex. So you go through different steps, right? And all of these steps can eventually be regulated. This makes it difficult. So what we did is we tried to serve the community by providing a lot of structural details so that we can, really find out how does the transcription machinery look like at these different stages and how are these complexes interconverted. And by now we have, also steps in between that weren't so clear 15 years ago, because knowledge increases, and we have a lot of knowledge on that. Now, I start at the beginning. You need to first free the DNA of the nucleosomes to make it accessible. And how do you do that? There's pioneer factors that can actually use binding energy to distort the DNA and to partially unwrap nucleosomal DNA. So that was exciting, to see the first structure of a pioneer factor bound to a nucleosome. So you see there's some DNA unwrapped here. And this is the beginning of opening up chromatin. Because, when the chromatin is closed, the gene must be off because the proteins cannot even assemble there. The polymerase cannot even form a transcription initiation complex. And then these pioneer factors actually look like this. They have a structured domain which can bind the DNA, and then they have these large unstructured regions. And what do? They are like glue, and they are recruiting other large protein machineries to this site where you want to start transcription. And some of those are so-called chromatin remodeling complexes. And what do? They use the chemical energy of ATP to shift around the nucleosomes, or even sometimes to evict nucleosomes. And I've shown you these machinery, so we also solved the structure of these so-called switch-SNF remodeler type of enzyme complexes. And you see here a nucleosome, it's called the -1 nucleosome. And here you see another one, the +1 nucleosome. And these machines can shift those nucleosomes to the side because they pump the DNA in the opposite direction. So basically, you have two nucleosomes, the pioneer factor comes, the chromatin remodeler is recruited, and then you use chemical energy to move these nucleosomes apart. And this way, you can actually open the DNA around the promoter. And now the gene, in principle, can be transcribed because the RNA polymerase II initiation complex can assemble. And the polymerase is complicated, but the pre-initiation complex is really complicated. So you have in silver here the RNA polymerase, but then you have all these other proteins around. Actually around 50 to 60 polypeptides, depends on how you count. And what is exciting about this machinery, it can not only recognize the promoter, it can recognize where transcription can start, but it can also open promoter DNA, and then it can help the polymerase to start RNA synthesis de novo. Because remember, when you do replication, you first need to synthesize a small primer and then you start. But in transcription, you start de novo. You just open the DNA, you separate the two DNA strands, and you can start the synthesis of an RNA chain. That's why you need such a complicated machinery, which also can be regulated. And what is also interesting, maybe you see that no matter whether you use or solve the structure of yeast, so single cell eukaryotic machinery, or the human one, they look very similar, at least to somebody who's not thinking about transcription all the time. So very high conservation of that machinery. It was invented once in evolution, and then of course, more proteins were added, but the basic machinery is highly, highly conserved, which also tells you something about its importance. I even think that at the heart of the polymerase, you have the first protein. And why do I think so? Because in an old RNA world, that protein was a replicase for RNA. And later, when we switched from an RNA world to a DNA world, that protein acquired the ability to also use DNA as a template. First, it only used RNA. And actually, we published about 20 years back that the polymerase that we have in our cells, it's so conserved that it can still use RNA as a template. It will not make long products, but it can still make short RNAs based on an RNA template. So I'm pretty sure also I can never, prove this, that at the heart of the polymerase is the first protein ever, which was important for the transition from the RNA to the DNA world. And ever since, the machinery grew, because you needed more and more regulation, to make different cell types and so forth. But at the heart of the machinery, the catalytic site that is really, really ancient catalytic site. Okay, now you probably thought, oh, it's complicated, but it's only the beginning, unfortunately, because the inside of a cell nucleus, actually we looked at it today, it's all black, you don't see anything. When you look with the electron microscope, it's so dense, you know, the genome is heavily packaged into chromatin, it's full of proteins. So there's a great artist, David, and he thought maybe it looks a little bit like this. I think this is quite close to the truth. One day we'll see it. But basically what it means is, in this environment, you have to do the job. Can you imagine? You have to find the gene, open the DNA, regulate the gene. Not easy. And the reason why I love this image is, you see here the nucleosomes, we were just discussing them. People think the nucleosomes are there because you need to, you know, wrap the 2 meters of DNA into this micrometer size nucleus. Yeah, that's one important point. But there's also co-evolution of chromatin and transcription. Because obviously you play around with chromatin, and then, you know, obviously you can use it to switch genes on and off. I just showed you the first example. When the promoter is covered, no activity because the polymerase cannot get to the promoter. And then you have all these factors that are used to remove the nucleosomes, to shift them away. So you have an additional layer of regulation. And because I want to show you new data, this is why I want to focus briefly on this co-evolution of chromatin and transcription. Right? I'll give you a few examples. If you look at and now we see that there's a difference between yeast and human. What I showed you so far, you saw the very similar machineries, right? But because the regulation in human cells needs to be so much more sophisticated, a lot of it is chromatin-based, and you don't have it in yeast. So let's look quickly at yeast. So we see the +1 nucleosome, which is the first nucleosome in the gene, you know, just after the transcription start site, about 60 or so base pairs down. That's the diet position. And here's our transcription pre-initiation complex. And so we solved the structure, and that was actually the first structure of a pre-initiation complex with the nucleosome. And we saw that the pre-initiation complex can dock to the nucleosome like this. And actually it's not a specific interaction. It doesn't recognize it. It's using it as a ruler. And that solves a very old problem. I don't know, who knows the Tata box? Who has ever heard of the Tata box? Many, yeah. But what many people don't know, that only 10, maybe 12% of the yeast genes have a Tata box. So you open the textbook and say, oh yeah, you have a Tata box and this is how the polymerase knows where to go. Yeah, but what's about the other 85, 90% of the genes? The proposal is, because you position a nucleosome, you can use that as a ruler to position the pre-initiation complex, and then you can initiate it in the right region. And then you can, you know, find the start site even if you don't have a Tata box. And that seems to be what's happening. Now, if you now go to the situation in human, then you see something very, very different. So in human, you have different promoter elements, and it's, you know, you also have a different set of general initiation factors. So it's more complicated. The mediator looks different, has 10 additional subunits. And then when you look at the +1 nucleosome, you see something that's very different from yeast. And what you see is that the +1 nucleosome, with respect to the transcription start site, can actually move. So in yeast, you always find it at a certain distance, right? And here, in human genes, you find it at different sites. And then what you see, and this is the advantage if you have the in vivo and the in vitro approaches, you see that when the nucleosome gets too close, the transcription is actually downregulated. Right? So you can reduce transcription activity by putting a nucleosome close to the transcription start site. So you move here from 18 to 0 distance, and you see in vitro it goes down. And also this is the other direction. You switch off the gene when the nucleosome is too close, and this is in vivo, using all the genes in human cells. Now we solve the structure of both states, when the nucleosome is distal and when it's proximal, so far away or close by. And when it's distal, you see there's space and you have an active transcription pre-initiation complex. And when it's proximal, so you move the nucleosome closer, you dislodge this part of TF2H. This is actually an ATPase, a translocase that you need to open the DNA. So this is moved out because the nucleosome competes for it. And you see this conformational change here. Just unbelievable. This domain, it's also part of TF2H. Suddenly you fold part of it into this long helix. And we know that this is an inhibitory conformation. So it's a beautiful example of co-evolution of chromatin and transcription regulation. So basically a factor evolved, TF2H, that can change conformation depending on how close the nucleosome is. Just unbelievable. It's co-evolution. And so basically by putting the nucleosome closer, you can switch transcription off and you induce this kind of repressive state of the TF2H factor. So basically in human genes, nucleosomes are at different positions, but very, very frequently when you look at active genes, you find the +1 nucleosome at 140 base pairs downstream. Remember, yeast was 60, and now suddenly it's 140. How can that be? This is for many active genes in human. The reason is that it's not next to the initiation complex anymore, it's actually next to a paused polymerase. And for that, you need to know that in human cells, you have an additional layer of regulation, and that is promoter proximal pausing of polymerase. So you have the pre-initiation complex, you open the DNA, polymerase starts, but it doesn't get very far. It pauses again at about 50 base pairs. So the RNA is very short, just 50 nucleotides, and then you pause this polymerase. And it's interesting, it's pausing just in front of the nucleosome. So we tried to get this entire structure, and we got it in several pieces so we can make a model. You see here the pre-initiation complex, the paused polymerase, and the nucleosome now in front of the paused polymerase. Here's actually the negative elongation factor. That's why it's red light, right? A red light, red traffic light. It's because it's stabilizing this paused state of the polymerase. It needs to be removed for the polymerase to start elongation. Now, we recently published, actually I'm not sure if it's out yet, yeah, maybe it just came online. We looked at the question, is actually the nucleosome involved in pausing the polymerase? Or is it just by accident, they are next to it? And it turns out that nucleosome contributes to the pausing. There was a big question in the field, is it the DNA sequence, this, that? And what we did is we depleted very rapidly a factor called FACT, for facilitates chromatin transcription. And what you see after 4 hours of depletion is that when you do MNA-seq, that these nucleosomes, these are actually the nucleosome positions. So that would be the +1 nucleosome. You see it's decreasing. So basically that nucleosome is becoming unstable. It's not present anymore, not the occupancy is low. And then when you ask by MNet-seq, it's another technology to actually map the promoter proximally paused polymerase. How much polymerase do I now have? You see that about only a third or so is paused. So that +1 nucleosome is contributing to the pausing. So +1 nucleosome contributes, the negative elongation factor contributes, and what you need to do if you want to now make this polymerase move down the gene, you need to use a kinase complex. You phosphorylate these proteins and you bring in other proteins. So that's a major switch. It's yet another layer of regulation. You take the paused polymerase and you convert it into what we call an activated elongation complex, EC star, different proteins. So we looked at that switch and we know exactly what the mechanism is because we have the structure of the paused and of the activated elongation complex. And without going into details, one thing you see is that you can either bind the negative elongation factor in the post complex, or you bind this PUF1 complex in orange in the activated complex. The two are mutually exclusive. So that's the most simple switch you can imagine, right? You either bind the negative factor or the positive factor. And the switch is regulated by phosphorylation. So then, transcription factors may bring this kinase. Is something that is currently being studied. We don't know for sure how that works. But once you bring the kinase, you convert the post complex into the active elongation complex, and then the polymerase can go through the gene. And now we went in vivo and we checked, is that true on all genes? So we inhibited the kinase. You can do that using the so-called Shortcut mutation. So we give a chemical to cell lines which carry this Shortcut mutation, then you only inhibit this kinase, which is called CDK9. And then what happens, we predicted, of course, that now the duration of pausing is longer. And this is the pausing duration of the polymerase, 2 minutes, 4 minutes, 6 minutes. And you see that every dot is actually a human gene here. You see that the pause duration is longer. But what was really striking and unexpected is what you see here, is that actually the initiation frequency, so how often you start the polymerase, is going down quite dramatically. How did we find out? Because we developed this kind of multi-omics approach. So we combined different omics methodologies, and then we use kinetic mathematical modeling to fit the data, and then you can extract kinetic parameters. And remember from your chemistry, if you want to get the mechanism, you have to have the structure of the intermediates, but you also need to know the energy barriers, and the energy barriers are related to the kinetics, right? Remember activation energy? So this is what we can do now. We can go into cells, we can do the kinetics, and we can look at our intermediates and see how they are connected. And what we found, and it's always interesting when you find something unexpected, is that also we are here controlling elongation, we are controlling the pausing release step, right? We are controlling elongation, but we have an effect on initiation. This was very unexpected. And this solved another very old question, namely, how can it be that genes can be regulated at this pausing step? Because the average human gene takes 40 minutes to transcribe. So it shouldn't matter whether the polymerase pauses 2, 3, or 4 minutes, because it means that it's transcribed after 39 minutes, 40 minutes, 41 minutes. Doesn't matter. So how can you control genes at the pausing step? The answer is that you influence the initiation rate, and then that means you can have 2 times, 3 times, 4 times, 5 times the number of mRNAs. Suddenly you get huge regulatory potential. Okay, this is the activation, but then something happened in the community. We thought, oh, now we are close to understanding how it works, how you regulate the gene. And then something happened. Several laboratories found that this post-polymerase is not as stable as you think. Often it falls off. So you get what we call early termination. It gets even more complicated, because now, it depends on how often you initiate and how often you terminate, if you want to know how many polymerases make mRNA. And that's what we want to know for transcription control. So, okay, there was a student, and he told me, I want to have a really, really big problem. I said, okay, we have a really big problem. How does that work? And this is Isaac, and believe it or not, he solved this problem single-handedly, starting from the cloning. And this integrator complex here has 16 subunits. Nobody ever made it. His PhD was to make it, and then his postdoc was to get all the structures. And now he has 4 offers for professorships in the US. Just a few weeks ago that he told me this. So it's unbelievable. He solved this problem. Here's the termination complex. And if you're interested to see how it works, it's I would be scared, you know, if I was the polymerase, I would be scared of this integrator complex. Because first of all, it can grab the post complex like this, and then see it's resolving this green protein, and this is actually the protein that makes the post complex stable. So it gets rid of this, and it also cleaves the RNA. You see, there's an active site in an endonuclease, which closes when it binds the polymerase, so the RNA is cleaved. You don't want this, because now the cap structure is cleaved off. Remember from your old biochemistry, the 5' end of the RNA has a cap structure to protect it. But this complex will cleave it off. Not good. It's another way to destabilize it. And because this is such a dangerous complex, it also has an inactive state, because you don't want this to be active in the cell nucleus all the time, for sure not. But Isaac's work was great, and again we had the mechanism, but again we had to ask now, is that really happening in cells? And this is what I want to show you. It's actually unpublished work, but it's basically done. It's close to coming out. We used a transdifferentiation event to check whether early termination is also used for regulation. Remember, now we said initiation is used, but also elongation is used, the pausing. Both can regulate. Now what about early termination? Is that a third mechanism that is used by cells? So you express one of these so-called pioneering factors, these master transcription regulators called CEBP alpha, in B cells, and then after a few days you get a totally different cell type. So you switch cell identity, it's called transdifferentiation. And this is such an interesting model because you totally reprogram your genome, and you have now different gene activities in the cell. And you can use that to study gene regulation on a global scale, going from one cell type to another. And what we found is, and that is for a subset of genes, so not all genes do that, it's a subset of genes. We found that indeed termination is used to regulate genes. So for example, one group of genes can be downregulated not only by reducing initiation, but also by increasing termination. So you know, in B cells or pre-B cells, there's hardly any early termination, so the gene is on. But then in these macrophage genes, the polymerase that is paused is terminated. So it's part of the regulation. Now, can it also go the other way? The answer is yes. We found a different gene group that is doing the opposite. Instead of increased termination, we now have reduced termination. So you have these genes are naturally terminating at a high level in B cells. And now when you transdifferentiate, this termination stops and the polymerase moves along the gene, so the gene is on. So what we found is there's a third fundamental mechanism how you regulate human genes, and that is at the level of early termination. And if you forget everything about my lecture, then please remember that slide. Only this one slide. Then hopefully you learned something. If you want to regulate a gene, right, you need to make more or less mRNA copies. This is how you regulate a gene. It doesn't matter whether the polymerase sits here for a minute or not. The only thing that matters when you look after a few hours, how many mRNA copies did I make? Right? Very important, because there's a lot of misconception, also in the textbooks. How many mRNA copies do I make per time? That is the gene activity. And they have to be complete, right? If you don't have a cap, you cannot translate. If you don't have a poly-A tail, you cannot export, and so forth and so forth. So complete mRNA copies, matured, processed. So how many do you get? That depends obviously on the initiation rate. You know, when you start more frequently, you can, obviously more polymerases will make RNA. But it also depends on this promoter proximal pausing, because when the polymerase sits here, 2, 3, 4, 5 minutes, it will stop initiation. And that is, you know, how at the elongation stage you can regulate genes. That is what I showed you, you know, with this complex with the negative elongation factor and the kinase that then is needed to activate the polymerase into this transcribing complex. And now the third way to regulate is early termination. You know, obviously you all know the classical termination pathway here, which is coupled to the polyadenylation. This is how you end transcription. This is, when you look at textbooks, that's how termination works. But there's also the possibility to terminate this early post-polymerase, and that also, in a way, changes the initiation frequency. Not the actual frequency, but the productive initiation frequency. Because when you initiate 10 times, right, and these polymerases are paused and stable and then go on, then you have 10 polymerases making 10 mRNAs. But when you initiate 10 times and every second polymerase is terminated, you will only have 5 polymerases go to the end. So only half of the messenger RNA. Now I didn't tell you how these 1,600 transcription factors do their job. And I think it needs another generation of scientists to figure out how these different types of regulation are used by these many different transcription factors. We don't know. And remember, on top of that, you have the chromatin regulation. So we will see, you know, what these transcription factors do. I'm sure some of them can do all of the jobs. And those are probably the master regulators, but I don't have evidence. It's just what I imagine, because nature will do everything it can, right? It will, at some stage, explore the different possibilities. Now I'm looking at Pavel, 45 minutes, whether I still have 5 or 10 minutes, or whether, yeah? Okay. Everybody's okay? Then I will show you another movie, which is quite interesting, I think. Now imagine the polymerase went through all these different stages, and now finally you have this actively transcribing complex which has all the right positive factors. Everything is fine, it didn't terminate. Integrator is shielded off, fine. Now these polymerases go through the gene very quickly. Right? In 1 minute they will synthesize about 3,000, 4,000, 5,000 nucleotides. So they go through 10, maybe 15 nucleosomes every minute. Can you imagine? Go through the nucleosomes like this. And by the way, the histones are generally not lost, because otherwise if you would lose the histones by the polymerase going through, you would lose the information that is stored in the post-translational modifications of the histone tails. You don't want that. So not only does the polymerase go through at a high speed, through chromatin, you also need to preserve the histones and reassemble the nucleosomes as the polymerase has gone through. We have started to analyze that process, and I show you some results. First of all, in vivo, you can find out which proteins are needed, right, for a polymerase to go through nucleosomes. For example, you need a protein called SPT6. Just to show you, this is the factor here, SPT6. You need that because when you deplete it, you know, the polymerase that normally goes through the nucleosome gets stuck here. This is the occupancy of polymerase inside of the first nucleosome. So normally it pauses beforehand, I've shown you that, and then it goes through quickly. But when SPT6 is lacking, it gets stuck here. You have higher occupancy. So you can find out in vivo which proteins are important for the polymerase to go through the nucleosome. But then you can also do the biochemistry again. And this was another PhD, Lucas, who's now also a professor at Harvard. And this is how he did it. He put the polymerase at the end of this nucleic acid scaffold, and then he runs the polymerase through the nucleosome in the test tube. And now, of course, you can add proteins, and you can see whether it goes through the nucleosome or not. And you get interesting gels, and you find a lot of factors that you actually need for the polymerase to make a full-length RNA. You see in the beginning you don't get any full-length RNA, but you add more factors and you can get full-length RNA. So when you have these data, then you can solve the structures to understand how these proteins help the polymerase to go through the nucleosome. For example, CHD1, it's using ATP hydrolysis to pump the DNA into the polymerase. So, you know, polymerase gets stuck in the nucleosome, then CHD1 comes and pumps DNA in so that it helps it to move forward. This factor I already mentioned facilitates chromatin transcription. It's more like a chaperone that keeps the histones around so that the nucleosomes reassemble. And I told you, you need this for the polymerase to pause. So the idea is, you know, one polymerase goes through, and then FACT reassembles the nucleosome, next polymerase can pause again. And you need the pausing to exchange the factors and then go on. Now, first movie of how the polymerase goes through a nucleosome. Of course, this is very preliminary and it shows only a few things and it's much more complicated. But you see it's a competition. The polymerase is a strong motor. Actually 20 piconewtons of force people have measured. But the nucleosome is also very stable, right? The DNA wants to be wrapped around histones. But at some stage, one of the histone dimers, H2A, H2B, is lost because the polymerase is moving forward. And now it's almost gone through, but the nucleosome tries to reassemble itself. So sometimes the nucleosome wins, and we've trapped that also in a structure I show you now. And then everything runs backwards because now the polymerase cannot progress anymore. And the nucleosome wants to rewrap. So you see it's going backwards, backwards, backwards. DNA is going backwards, RNA is going backwards. And now the RNA is actually moving out of a pore underneath the active site. Isn't that exciting? And then we have this cleavage factor that gets rid of that RNA. That means the polymerase gets restarted and it can try again. So the nucleosome will never win because there's this yellow rescue factor called TF2S. And you know, whenever the polymerase gets stuck by backtracking, this factor comes and restarts it and the polymerase tries again. And you know, you try as often as you need to go through. And in the end, it's a thermodynamic process. Eventually, the polymerase will always go through the nucleosome. That's what I wanted to tell you today. Remember the first slide I showed you with the different intermediates. Now I think you know that kinetics is important to understand how you interconvert these intermediates. And just to mention this again, you always regulate initiation because that determines how many copies of mRNA you make per time. And of course, these 1,600 activators are important, but the pausing, the elongation, and termination can feedback and change the rate of initiation, right? Because not all of the polymerase that initiate make it to the end, because some of them are terminated, or because when you pause longer, the initiation rate is actually dropping, so you can change how many polymerases actually initiate. Summary. I think this is fair to say that, you know, in contrast to 30 years ago when we started as PhD students, 1994 was the selection, 1995 was the start. So 30 years later, I think it's fair to say that many aspects of the transcription mechanism could be elucidated using structure-function analysis in vitro and in vivo. And it's also fair to say that three fundamental mechanisms now emerge that you use for transcription regulation. And those need in-depth analysis. What do I mean? We don't know. How does that kinase that is so important, how is it recruited, right? What exactly the histone modifications do? Are they changing the affinity of certain factors so that it's less or more likely that you get termination? There's many, many questions. So this needs in-depth analysis. And I think the dynamic interplay between chromatin and transcription, the co-evolution, you know, how chromatin helps in transcription regulation, this is becoming a little clearer, but really it needs a generation of people to investigate that in detail. I showed you some very prominent examples, but we will probably see very interesting, you know, proteins that can alter chromatin structure and thereby indirectly regulate transcription. I'm pretty sure that this is something that will come out. We have evidence already that some of that exists, and there will be more to come. Now, because this was an overview, of where we stand now, it's very difficult to mention individual people. I mentioned Isaac because he single-handedly did this recent integrator project, but we had many such wonderful people. I mentioned Lucas briefly because he was the first to get a system where polymerase and the nucleosome are together in a, you can use them together in a test tube. But we had many wonderful people. I just see Sandra here. She did the pre-initiation complex. She made recombinant TF2H. So each of them have a story. And you know what gives me great pleasure? I just did a travel through Asia because my new job, it's also about international collaboration and science policy. And I come to Seoul in South Korea and I see Jinmi. And she's a professor there now. And I go to Tokyo and I see Kayo. I don't think she's here. She had left already. And she's a professor in Tokyo. And that, of course, gives me great pleasure. The field goes on, but also the individual people who worked really hard on the problem now can live their dreams and do their own science. And that is really wonderful. Yeah, and that's my new portfolio. Instead of 84 genes, it's now 84 institutes. And I tell you, it's incredibly interesting because the Max Planck Society has institutes ranging from astrophysics all the way to the history of art, for example, in Rome. And that actually brings me back to the beginning when I mentioned Mendel, because do you know that he published 13 papers? 13, 1-3. 2 of them are on botany. And they were apparently quite influential. And then the remaining 11 were on astronomy. For example, he described phenomena, weather phenomena, a storm, for example, he described. And we just saw here the telescope about this size that he used to observe the universe. And I think that's a science, right? We go into the unknown because we're curious, and then we have to cross boundaries. We have to bring fields together and think outside of the box, and then we can do what is called disruptive science. And I wish that many of you have such moments in your career, or maybe some of you had them already. I had one at Stanford at the time when I knew I'm going to solve this problem, how the black box can be opened. And when this happens, you immediately know you will not forget your whole lifetime. This is changing your career. And I really hope that many of you, because I see many young people here, will have such moments. And with this, let's see, yeah, we are really at the end. And I would thank you again for being such a great host. Of course, Pavel, but there's a whole team behind it who organized. And we had great visits today at the 2 big universities here. We saw great facilities, from nanoscience all the way to molecular biology. Very motivated people, because it's just one thing to have the equipment and the money. The other thing, of course, always is to have the people and the ideas. More important even. I was told at Stanford, it doesn't matter, how you do the experiment. It matters which experiment you actually do. So in the end, it all happens in your head, because there has to be a concept, a hypothesis. There has to be some plan in your head. And then it's important not to keep on to it, but to allow for it to be modified. 10 years ago, suddenly termination. Everybody thought the post-polymerase is stable, suddenly it falls apart. You have to allow for this to discover something new. And this is what I wish. Thank you so much. It's really very moving to be in the refectory where Mendel used to eat. And yeah, I don't know if you allow for questions, but I would like to thank you very much for your attention. Thank you.
Patrick, thank you so much for this fantastic lecture. Very illuminating. Lots of topics. This is a scientific lecture. You will be grilled now. There will be questions, right? So I open the discussion in the audience who has a burning question about transcription or regulation.
Hi, thank you for your fantastic talk. I have such a question that I think that now the general view is that transcription replication conflicts are a sign of cell metabolism dysregulation and are actually not a good thing for the cell. But when you speak of premature transcription termination as a way of regulating transcription, could this be kind of also a way how to stall and eventually terminate transcription on purpose? Or do you think it is an overkill mechanism?
Yeah, absolutely. I think it's a great question. First of all, I think transcription replication conflicts should be studied. I know it's difficult because also they're related to cancer progression, because you can induce, as you know, DNA breaks. But to your question, you know, the integrator complex is such a new protein that there's few papers. And we've done one collaborative study with Torben Haig Jensen in Aarhus in Denmark, where we saw that it's a general factor that is used at many genes. But we also saw that it's the point where it acts is not as defined. So today I was oversimplifying. So it could well be that integrator actually can not only be important for a regulation of transcription, but it could also be this kind of surveillance or factor, you know, that can take care of stuck machinery and remove it from the genome, maybe as part of a repair or a surveillance pathway. This is actually quite likely because many of these proteins have different jobs, right? And it's just because we scientists focus on the problem we are interested in. I don't know if you know this analogy. Somebody comes a bit drunken from the pub, right, and then gets home and cannot get in because has lost the keys. And then he goes back and he only looks under the street lights because it's the only place he can look. And it's a little bit, in science, it's a little bit like that, right? Because we are, you know, that's why genomics is good. Instead of looking what's out there, we always try to see what we expect. And this is why serendipity is less frequent, because people fail to see, and this affects everyone. So I would not be surprised if somebody, you know, does genome-wide studies of integrator under different conditions, where maybe the replication is getting stuck or so, that you would see such a function. It would be a wonderful PhD project. Clear design, right? You change the condition, you do genome-wide mapping of the integrator, and maybe you find exactly what you suggest. But I don't know that, I don't think there's any papers on that, as far as I know. But I didn't read over the last year, I had to read other things, so maybe it has been done.
All right, do we have more questions? Yes, please.
Hello, I was just having one question to the TF2S that you were showing with the, like, you know, allowing the backtracking and then the restart. So did someone looked at it, for example, in the anomicities of splicing? Because there clearly the polymerase speed would have an impact, and if the nucleosome is slowing it down, then it could have a regulatory moment, right?
Not that I know of. It was actually a factor discovered already in the 70s, but I don't think a role in splicing was ever found. And there's old, you know, TF2S mutations in yeast, so it would probably have been found if there was something. I don't think there is. I don't know of any paper.
So I have a question which is kind of coming back a little bit to the approaches which you used and how you reinvented the way you looked at transcription. You know, it's very clear that structural biology and genomics kind of combined helped. What I was not hearing in your lecture much is imaging, you know, looking at dynamics and really seeing the dynamics of transcription and measuring it and looking at the biophysics of it. Can you see ways how this could be integrated into the picture?
Yeah, it's a really good point, Pavel. I think it's the future. I mean, one is, of course, tomography, to get structures in the cell. This will be very, very difficult, but, you know, if I wouldn't have taken the job I took, for the next 10 years, I would have tried to something like that, to develop tomography for nuclear biology, which is so far not really available. But what you say is equally important. you know, this high-resolution light microscopy, because there you are in living cells and you have real kinetics. And several people have started to do that, as you know. And I know that Stefan Helm, our colleague at Göttingen who invented stat microscopy, that he's very interested in looking at kinetics with high-resolution light microscopy. So we should have all the tools. We can put tags in, right? We have the microscope. So that would be another thing that I would have been extremely interested if I would have time now. But I hope that many of the young people go in this direction. Because again, you can do kinetics, right? You can do depletion experiments, all of that. And you can test your hypothesis in vivo.
So I think with that, I would like to wish you that after you do the service to Max Planck Society, German society, and humanity, you can come back. You can come back to research and, you know, reinvent yourself one more time and use some new technology we don't not even know yet about. Okay.
Thank you, Paul. Thank you, everyone. Thank you. Thank you.